Good afternoon, everybody. Thank you for taking the time to come out here today. This project is between myself and Shoaib. Put your hand up, Shoaib. Yep, he's my right-hand man and left-hand man and everything else. So it is a 5-year national collaboration that Horticulture Innovation have co-funded. And the whole premise of the project is to increase pollination services. So they have a range of insects for different crops at different regions and in different climatic zones. So, whether it's bees and a fly that I've got circled there, basically this is a industry-driven demand that Horticulture Innovation's identified 6-7 years ago, put out a request for someone to look at that. And the reason it's such an issue is that bee numbers are declining worldwide. It doesn't mean they're actually going backwards, but the amount of crop that's being planted that requires bees as a pollinator is increasing at a faster rate. So the gap's getting bigger. And also, sole reliance on one insect species to do all your pollination is fraught with danger. And the varroa mite incursion last year was probably a good reminder of that. So the reason bees are declining is, look, it's a combination of environmental contamination, whether it's pesticides, pollution, microplastics, herbicides, glyphosate, global warming, particularly habitat fragmentation. So a lot of native landscapes have been broken up into small bits. So you have small patches of remnant vegetation, and as some of our previous speakers, Josh and Steph, have highlighted, that obviously decreases your biodiversity. And then you have a loss of alternate flower sources. So bees certainly don't like to just visit one plant or one flower, and in particular avocado flowers. So every time we turn up down at avocado farms and they've released bees, they said, we're just letting you know they're really angry because I've woken up in an avocado orchard. So they don't have any choice. It's not the best, obviously it's not a great crop for honey production. So literally they're providing the bees just for pollination needs. So they will go off if there's alternate flowering plants nearby. So why flies? This is one of my favourite flies. I was talking with Josh's relatives, who obviously did most of the work for his project. Is it Erin? Yep. Erin said, I'm interested to see if you can make a talk on flies entertaining. I said, trust me, by the time I've finished, you'll be wanting to talk about flies the rest of your life. So this is a sarcophagid. Sarco means eating flesh, which is a wonderful thing. So we know that flies do play a role in pollination. They're the second most important and most prevalent visitor of flowers after bees. There's plenty of evidence in the scientific literature. This goes back to Brad Howlett in 2017, who looked at pollinating insects of avocados and macadamias in the tri-state region of Australia. And we actually did a literature review that got published in Insects that talked about all the flies that are involved in pollinating crops in Australia, and there's an awful lot. And we actually reduced the number down to about 12 species. So there's about 6 caliphorids or blowflies, 3 or 4 hoverflies, and 2 species of rhinid flies that are called snout-nosed flies. We don't have them over here, so don't look around for a snout-nosed fly. And then just very recently, and again this is supporting the work of Steph and Josh, a paper came out about providing alternate sites for dipterans, whether it be syrphids, hoverflies, droneflies, to lay eggs in an agro-ecosystem around your crop. This is one of my favourite flies that we started off with, Calliphora albifrontalis. It's native to southwest WA, and as you'll note, it's a very hairy fly. So most of the flies that we work with as pollinators are big flies, and they're very hairy. They regularly visit flowers. They'll visit probably 100 flowers in a row in succession because they need the nectar or the sugar to be able to fly. So it's just an energetic demand. And they're accidental pollinators. They just pick up pollen and move it to the next flower. So what are the advantages of having a fly as a pollinator? Firstly, there's flies around all the time. Any time of the year you'll find some sort of fly. They can typically operate at cooler temperatures. So that fly in that picture is Calliphora vicina, which can operate at 5 to 10 degrees, no problems. They don't sting workers, which is always a good thing, and they're cheaper to rear. So what flies are feeding on avocado flowers in southwest WA? And this is good to link in with Josh's work, because he's saying, okay, what insects visit the flower, but we particularly looked at those that fed on the flower for a considerable amount of time. So when we started, a bit like the hoverfly invasion, we had thousands of honeymoon flies, as you can see why, or love bugs. So they're a small bibionid fly. They're not a suitable managed pollinator at all, because the adults only live 5 to 6 days. So you'd be releasing eternally. So that's Albi frontalis. As you can see, the dots there indicate its distribution in Australia. It's very localised. Clifford dubia, the western blue-bodied blowfly. Now, we've all used the expression running around like a blue-ass fly. Well, that's where it comes from. So when you watch this fly, it's got a really erratic flight path and it just keeps changing direction. So think about Clifford dubia next time you say that. That's found over most of Australia, and then Clifford vicina is found in the southern half of Australia, so it likes cool weather. Some of the hoverflies we got, you've probably seen a lot of these, Melangita viridiceps, you don't have to remember these names, although I will question Josh at the end of this. That was the one that was mainly attributed to the hoverfly invasion of 2020, and a very small one. So to know how effective a pollinator a fly is, you need two things. A, you want it to be abundant in the sense that it does lots of flower visits. And then when it visits, how many pollen grains does it deposit on the stigma of the female flower? So we actually did some, it's called SVD, so single visit deposition of pollen. We did it with a couple of flies. And you can see we've got Clifford dubia on the left here. That's the mean feeding time. So this is feeding time first, which is 30 seconds, and Clifford vicina was very similar. So it's quite a long time visiting and interrogating a flower. It's probably not the right word, but you know what I mean. And then we link that in with how much pollen do they actually deposit. So on the left we've got bees in red, and these are 3 blowfly species. So you can see that Clifford vicina was depositing over 1 pollen grain per visit, and bees were sitting at about 1 as an average. And considering that an avocado flower needs multiple visits to get enough pollen grains to stimulate pollen fertilisation, it's really important. So just a single visit by any insect will not do it. And then if we look at hoverflies, so there are those 2 hoverflies I showed you before. Most of them are less than 1. This fly here, Eristalis tenax, which has been mentioned, the drone fly, that again was up around 1.66 and again higher than bees. Okay, so what flies did we test as pollinators? So clearly based on that data I just showed you, they're the 3 species we looked at. The southwest WA fly, Australian fly, and that one's found globally. So it came into Australia about 100 years ago, along with cane toads and everything else. So the first thing we did was we did paired tree enclosures. We didn't know anything better at the time, so we put a type A and a type B tree together, or they were next to each other. We just enclosed them, and that you can see is an aerial view of where the enclosures were throughout the orchard. And I must give a big thanks to Jasper Farms and all the staff there who helped us put these things together. And the first thing we did was we said, okay, let's just put this one fly in, Clifford albi frontalis. And then we had 9 enclosures where we just excluded insects to see what would happen. Now it seems kind of obvious that you're probably not going to get much fruit, but we thought let's make sure we cover that off. So once flowering had finished, dismantled the enclosures, went back, counted the fruitlets, and followed it through to mature harvest. So that first year we actually showed that if you don't have any insects, you literally get no fruit. So that again reinforces the need for an insect vector agent to assist. Albi frontalis wasn't great, but it certainly showed that even at final harvest, it was about 30% less than what you got in trees in the open that obviously had bees and any other insects in the orchard helping with pollination. So we got all excited, and the next year we did, again, paired trees, but this time we used 2 fly species, albifrontalis and dubia, and we had 500 flies in each. So again, this is the first fruitlet count, lots of fruitlets in the open, but by the time we got to harvest, Calliphora dubia was about 2/3 as good as the open pollinated. So remember, we're not trying to find a fly that is better than a bee. It's probably very unlikely. You want something that's going to give you some benefit, obviously, in the long term. So that was pretty encouraging. So that's the key fly that we think is doing a really good job. And that actually fitted in with, we actually did some flower feeding observations. We got a whole lot of cages of albifrontalis and dubia, put in same number of avocado inflorescences, recorded visits, and then rotated them around all the time. And we got Calliphora dubia fed this one, oh geez, hang on. This one here fed 3 times more often on the flowers than albifrontalis, which kind of explains that difference in the yield result. So one of the questions we got asked was, well, how many flies are you going to put in there? How many flies should you put in there? So we tried to make it equivalent to the number of bees per hectare or per tree. So let's say the orchard was using 3.5 hives a hectare. That's 3.5 times around 45,000 bees. All the bees aren't out foraging at once, so we assumed 40%. So that's about 63,000 bees per hectare. If you then account for the tree density in that orchard, so it was 320, that works out about 200 bees per tree at any one time. So we released a similar number of flies. So then we went big scale. We went to large enclosures in 2020, and I think we covered 36 trees in total. So there was 33 hasts and 3 pollinisers. So we had problems with our flies at that time, so we just compared bees in an enclosure versus the trees outside in the open. And what we got, so we did this at 2 sites, so at Pemberton and at Busselton. You can see the trees in the open obviously had a higher yield than the trees that were just in with the bees. So we're trying to separate what bees did versus what all the other insects could do. And I'm pretty sure because of 2020, that hoverfly invasion, that difference we're seeing there was due to the hoverflies, just their sheer abundance. So that's them having a jolly good time in the orchard. And they don't spread a lot of pollen, but so many visits, it just accumulated. So we're basically, those other pollination insects in the orchard were contributing about 30% higher fruit set. So in the 4th year, we tried Calliphora dubia, Calliphora vicina, and again, a new hive of bees. And we did the fly releases. We released 5,000 at the start of flowering, 3,000 2 weeks later, and another 2,000. Just because blowflies don't live for probably 6 weeks in the field, maybe 4 weeks. So you're going to get declining numbers over time. So that's why we did that. So that's the vicina after we released them. They're all sitting around wondering where they were. That's one of them feeding on a flower. So you can see they get right in there amongst the flower. They're trying to get, so there's all these, what are those things called again? Declan, help me out. That sounds good. Nectaries, yeah. No, that's it. Thanks. Yeah, so they're going around all the nectaries. You see the fly literally rotates around the flower, getting all the nectar it can. And in the meantime, its underside is brushing the stigma in the middle of the flower. Thanks, Declan. That's cool. Yeah. And that's Calliphora dubia feeding on it. So this particular year was really cold and drizzly and wet. And what we got was, I think there was a bit of a thermal effect with the shade cloth, and the bees actually did pretty well. But you can see vicina, dubia and trees in the open were literally the same. So if you'd have just done nothing, you wouldn't have got much fruit. So we've isolated at that time of year, and with those conditions and with those fly species, we've got a reasonably good result. And we did the same thing at Pemberton. And Pemberton, we actually got quite a dramatic improvement in yield with Calliphora dubia. Now, the reason why, and I'll explain that, is that we actually ended up putting in a double density of flies. We didn't intend that, but when we got to the site, we didn't know how many trees were in the enclosure, so we had all these flies in a cage. You can't sort of let them out and go, look, just the first 5,000, thank you, okay, and then shut it. So we just let them all out. And that's why we got that result. So, year 5, someone's out. Okay, we just did that last year. Okay, we looked at, now the reason we've now gone to a double density, so we've done the same 2 flies, but an equivalent density to bees and then twice the bee density. And the reason we can do that is because the cost of rearing 1 million flies at that time was about 1/3 of the cost of having 1 million bees. So we could sort of double the fly numbers without it being ridiculous. We had a problem with dubia, so they're not on there, but Calliphora vicina, you can see by the time we get fruit drop, is again about half of what you get if you just had bees. Now, the reason Calliphora dubia is not on there is because their adult emergence was severely compromised because of the weather. So there were really cold nights, so it was 1 to 2 degrees minimums, and 78 to 80% humidity, and all the flies came out with crumpled wings. I think someone mentioned it before about emergence and having to harden up. You said that, to harden up, the lace wings. Same thing with flies. When they come out, their cuticle's really soft and malleable, but they need a couple of hours for it to harden up. And the same with their wings. They're meant to like inflate their wings. Well, they couldn't. So they all ended up with crumpled wings and they couldn't fly. So that was the end of them. So the lessons we learnt from the fly releases are, it's far better to release adult flies than it is fly pupae. The reason being, your pupal emergence can be compromised either from wasp parasitoids, and that's an example there of Zealanthus. You can see the little exit hole. So when Paul was talking about wasp parasitoids, that's what you typically see in a fly pupae. And also a combination of cold and high humidity. And Shoaib repeated those conditions in a laboratory and we got exactly the same result. Okay, the other thing to do and be mindful of, no matter what insect you've got, is to have regular introduction of your native fly species into the colony to maintain its vigour and genetic diversity, and in particular, flightability. So, when an insect emerges, is it going to fly and do what you want it to do? So, the things we've learnt since then is that we've actually been playing around with rearing up flies in huge numbers, and we're talking millions. We've got the cost now for rearing 1 million adult flies, which would actually be about 1.4 million larvae, because there's a little bit of loss or mortality through the life history stages, is about $850 for 1 million flies. So this year, we want to test out vicina at 3 times the fly density, dubia 3 times, and we also want to look at a drone fly, this Eristalis tenax, which has been mentioned a couple of times. And the reason we can go to 3 times is that we've now worked out the cost of rearing 1 million flies is less than 1/5 of getting 1 million bees. So there's been quite a bit of talk and chatter lately about drone flies or syrphids or hoverflies, whatever you want to call them. It's a very big insect, and it literally does look like a bee from a distance. And it's a very good lateral forager. So it's being produced commercially in Tasmania for a company that is involved in seed production. So they have 1 and 2 hectare plots of carrots and brassicas and that they need to get pollinated, otherwise you don't get a seed bank. And this fly is very good at foraging right across all the lines, whereas bees will tend to pick out single parental lines. So as I said, that's one of the flies on a carrot umbel. I think that's the right word. Another fly that we've seen at Cable Farms and is present around most of Australia is another hoverfly species, but it's called a lagoon fly, which is to suggest comes from ponds and marshes and that, is Aristolinus punctulatus. It's a magnificent looking fly, and we're going to be playing around with that one as well. Another syrphid that we saw at Cable Farms was this one, Ostalis copiosa. So they're really big, dramatic looking flies, and they're very, very flower specific in their behaviour. So just to finish up, just letting you know what's kind of on the horizon in terms of pollination. So this project I'm in now is finishing the end of this year, but there's a 10-year project coming up that's called a CRC, which is a Cooperative Research Centre. It's a Wallace institution, but it's a huge national project. It's just looking at pollination security, so it's about the whole gamut. It's about environmental conditions, environmental plantings, interrows, maintaining native vegetation to encourage biodiversity. It's talking about managed pollinator insects and then matching them up with crops and regions. So DPIRD has invested in that, $1 million over 10 years, and it enables us to look at more fly species and novel technologies to retain them in an orchard environment. So a lot of people say, yeah, well, your bees will go back to a hive, but where do your flies go? Well, we're doing a dispersal assessment this year as well to sort of get that answer. But yeah, every fly is not going to hang around in the orchard, so we need to work out ways to keep them in there, and we've got some options with slow release lure technology and things like that we want to look at. So that's all for me, folks, so thanks very much. Come on, Josh.
Hi. I've not seen a lagoon fly here. Is that just because I haven't been looking?
No, not at all. No, like I said, we've seen them, but not frequently. Okay. You know, over sort of a whole day of observation, we might have seen 6, 8 of them.
So would it be a good thing to build some lagoons?
I don't want to tell you to go and build a lagoon.
I'd like to. I've got plenty of wet
children end up in it. I can't see why not. I mean, anything you could do to encourage other insects, and especially syrphids and hoverflies that do breed in those sort of stagnant aquatic environments. Yeah, it's not a bad thing at all.
Yeah, good, thank you.
For sure, yeah.
David, do the fly moves around or far from the trees or the location of release as much as a bee that we talked before? Josh said that was going over 1 kilometer. Would they go that far as well? So when you release them, it's easier to keep the numbers close by or not?
Well, that's what we want to answer this year. We want to do 2 fly species releases from a central point, and then we have concentric circles of traps. Not that suck them into the trap, but will catch them with a low level of sort of attraction. So we'll see how far they do actually move over 24 hours, 48 hours and out to a week. We know kind of how long they live in the wild. But look, they certainly are going to move further than bees would, but I can't give you the exact answer because we don't know yet. But that's what we're going to look at. But these slow release lures would give off a very low level of decomposition volatiles, so dimethyl and trimethyl sulfide, that we can't detect, but the flies can detect. And that would hopefully keep them in that environment, but also draw in ones from the surrounding vegetation and bushland.
I've heard that growers have put dead sheep and kangaroos around their paddocks. Is there any benefit in that?
Well, I know, particularly mango growers have done it, I know. And there was actually a study was done on what they call stink stations, and they had them in mango orchards. So they had chicken and fish carcasses, spread them out, measured the fly populations and looked at the yield. They found more blowflies, but no increased yield. The problem is whether it's a you know, a patty of liver or meat meal or something, or a dead animal, whatever, you've got no control on what insects come to it, and more importantly, what actually develops out of it. So a dead body carcass environment is really intense competition, and you tend to get 1 or 2 species dominate and come out. And if they're not the fly species you want, and it's not a good sort of look around the orchard, yeah. But yeah, it has been done, definitely. Yeah, no questions, Josh?
Yeah, a small comment on that one, David. Definitely, I know of mango growers, they did it, and they said it was a totally detrimental effect on that, on the workers.
Yeah.
They definitely didn't want to work in that orchard.
No. Well, there's a thing been going around called a fly grow patty that an avocado grower came up with, where you put these patties out, you know, sort of 8 weeks prior to flowering, and all these flies will come to it and breed up, and then they'll all come out and help pollination. Well, we tested them out and they were pretty much next to useless, because you got no control, and this would be the same with a dead carcass, you got no control of all these predators that come in. So the eggs get taken off by beetles, the maggots get stung by wasps, taken away by big staphylinids. So you lose just about everything. You end up with nothing at the end of it.